Hello, everybody. Today I'll be going through the lesson one on Mendelian genetics. Mendelian genetics. What is Mendelian genetics? So we have so many people. We have so many people who talked about genetics, but the work of Mendel will be the focus for our discussion today. What is the significance of Mendel's experiment to the study of genetics? What is the importance of the experiment being done by Mendel on the study of genetics? So the words we're going to be looking into today, one of them is genetics. We will talk about genotype. We talk about allele phenotype, dominant, law of segregation, recessive, hybrid, homozygous, law of independent assortment, and heterozygous. All of these things we're going to mention today. The word segregation means separation. So when you say something segregates, means it separates away from each other. So let's say, for example, we have two chromosomes. They are close to each other before in the same cell. So when they segregate, the meaning is one chromosome becomes to found in one cell and the second chromosome in another cell. So this is an example of what segregation is. So it is a separation of allelic genes that typically occur during meiosis. So this process of separation usually happens in meiosis because meiosis happens in such a way that when you have a diploid cell, it breaks up into one. Let's say you have two N, it breaks up into N, she's going to give you a haploid. So this is the reason it says it happens in meiosis. So you're going to have two haploid from one diploid. So how did genetics begin? What started genetics? So genetics is being started by studying the passing of traits from one generation to another generation. And this is what we call inheritance. Inheritance is the passing of traits from one generation to another generation. We also call this heredity. So we can either use the word inheritance or heredity. Gregor Mendel actually did a great job in publishing his findings on the method of inheritance in a garden pea plant. So he used a garden pea plant, whereas he cross-pollinated the pea plant. So let's say we have pea plant, which is normally known to produce a green pea. Then you have pea plant, which is known to produce a green pea, and another one which is known to produce a yellow pea. So what did they do? The one always produce a yellow pea, and also other one produces a green pea. So every time they produce pea, it's always either green or yellow. So this is the pod. So the pea would be the small bean-like structure which is found in the pods. So this is parent generation. This is the parent generation for green, and this is the parent generation for yellow. So Gregor Mendel decided to cross-pollinate these two and see what's going to happen. So he cross-pollinated the pea plant, which normally self-fertilize. Self-fertilization means the green will fertilize the green to give green. The yellow fertilizes the yellow to give yellow. But once we cross-pollinated them, it combined the two gametes to have what we call a hybrid. So it combined the two gametes to have a hybrid. So he rigorously followed various traits in the pea plant he bred. He studied several features of it. He studied the color of the peas. He studied the position of the flowers, and he studied many other, the height of the pea plant too. So he studied so many. So Mendel began the study of genetics as a one, and that's why the study of the genetics, which is the science of heredity, was started by Mendel. One trait Mendel noticed was the seed color. So the seed color was the main bone of contention. So he studied the seed color very deeply. Some plants always produce a green seed, and while others would always produce a yellow seed. Some will produce every time a green seed, and some will produce every time a yellow seed. So Mendel said, okay, you know what? This one that would always produce a green seed, I'm going to take the pollen grain from one of them, and I'm going to put on the one that always produces a yellow seed. So he took a pollen grain from the one producing the grain and put it on the stigma of the one that produces yellow, or vice versa. So Mendel called the green seed and the yellow seed of the plant, the parent or the pea generation. That is to say, the plant which always produces the green pea every time, and the one that always produces the yellow every time, he called them the parent generation. So now let's talk about the F1 and the F2 generations. The F1 is the first phyllo generation. So the offspring of these P cross are called the F1 generation. So he crossed the P generations, that is to say, the generation producing the yellow peas and the generation of the one producing the green were being crossed together. So when this crossing was being done, so what they produce is called the F1 generation. So the F1 generation is a hybrid, which is that this is a parent generation. So the parent generation. So the two parent generations will produce a hybrid, which is going to be a mixture. When you say a hybrid, means it's a mixture. The mixture of the traits in the yellow and the traits in the green. So after the production of the offspring from the first cross, all the offsprings were yellow peas. So then he decided to cross two yellow peas and see what's going to have. So that produces the F2 generation, so which is the second hybrid generation. So this F2 generation is called the second phyllo generation. So as you can see, it says the second phyllo, the F2 generation is the offspring of the F1 cross. So you will cross two F1 offsprings together to give you F2. So what Mendel noticed is when it crossed these two, the yellow and the green, in the F1 generation, all of the offspring, everything was yellow. So then he started to worry, where is the green? Where is the green? The green was not appearing. So, but what happens is when it crosses these two yellows in the F1 generation, then the yellow green appeared in the F2 generation. So in the F2 generation, there was a percentage of green pea to yellow peas. So he had 1, 2, 3. So he had a ratio 3 of yellow to ratio 1 of green when he did the second crossing to have the F2 generation. So the F2 generation, for every yellow, for every green, for every one green pea, there is a 3 what? There is a 3 yellow peas. So this is what we mean ratio 3 to 1. Is that clear? So now the diagram has been shown here. So this is the parent generation, yellow pea and a green pea. So all of the first phyllo generation were all yellow. So when a cell fertilizes two, it had this. So to every one green, there's three yellows. So if you try to do this, you would see is ratio 3 to 1. So when it counted the number of seeds he had 6,022 yellow and 2,001 green seeds from the cross. So what do we know about genes? Genes usually appear or occur in pairs. So Mendel concluded that there must be two forms of the seed traits in the pea plant. That is to say, there must be two forms of the traits in the pea plant, and that each was controlled by a factor. The meaning is there must be a trait which is controlling the yellow, which is going to be, let's say, let's use the uppercase Y for the word for the gene in the yellow, and you've got to have two of these. So then the gene in the green will be this. Why? Because this is the one having the one which is apparent in the first phyllo generation, which is said to be what? Dominant. And this is the one that appears afterwards, which is said to be what? Recessive. Which that is to say it's hidden in the first phyllo generation, because the dominant would overcome it, it covers it up, and it's not been seen. So now Mendel concluded that there must be two forms of seed traits in the pea plant, and that each was controlled by a factor. An allele is an alternative form of a single gene. One gene, for example, the gene controlling the color, would have alternative forms, which is allele. One controls the yellow color, and the other one controls the gene, the green color. So there's always an alternative form of a gene. So the gene for the yellow seed and the gene for the green seed, they are different allele for the same gene. So the gene for the gene for color has two alleles. One codes for yellow and one codes for green. So dominant alleles control the trait that appeared in F1 generation. So the dominant allele is the one that controlled the appearance of yellow in the F1 generation. The recessive alleles were masked. They're not lost, but they've been hidden. They're masked in the F1 generation. So in Mendel's cross, the yellow seeds were dominant and the green seeds were recessive. So the dominance, when you want to model inheritance, the dominant allele is represented by a capital letter. We would model the dominance with the yellow with the uppercase, and the recessive would model it with the lowercase. So an organism with two of the same alleles for a particular trait is homozygous. For example, let's take the color, for example. The yellow color, if you have this allele, you have two same allele. For the yellow, we will say this is homozygous. So let's say you have the same two alleles for the green. So you say this is homozygous as well. So, but the difference between this homozygous is one is homozygous dominant. One is homozygous dominant because it's the dominant one with the uppercase, and one is homozygous recessive. Is homozygous recessive. So an organism with two of the same alleles for a particular trait, which is the trait is color. The trait is color. So we would say the homozygous. Heterozygous is an organism with two different alleles for a particular trait. So let's say the trait is the same color. So the trait is a color. So if an organism has two different alleles, for example, an organism has this and has this. It has two different alleles for the same trait. So we would say this organism is heterozygous. So it's heterozygous for the trait. In heterozygous individuals, the dominant trait will be observed. This will be observed. So the teacher said this will give a yellow what? A yellow pea. And the recessive will be hidden. This one will be hidden. It's to be masked. It's not going to be shown. So the P will be yellow. So let's talk about genotype and phenotype. Genotype and phenotype are two words we use just to let you know that genotype is the gene that actually causes what we see. And the phenotype is what we see, which is being caused by the gene. So the appearance of an individual does not always indicate which pair of alleles it possesses. If I say somebody being tall, I cannot really say because he's tall, he's going to have only the tall gene. He could also have a gene that codes for him to be short. But the only point is the gene I see, the phenotype which makes him tall, is a dominant. There might also be some other ones which is not seen, which is recessive. So a genotype is an organism's allele pair. So an organism allele pairs, that is to say the one we see and the one hidden, is called genotype. So all of the total allele pairs an organism has is called the genotype. The observable, the one we see, observable characteristic, or the outward expression of an allele pair is called the phenotype. Now, let's go back to the pea plant. The pea plant, we see some is yellow, and we see some are what? Some are green. So the phenotype for this pea plant is yellow color, and this one is green color. But the gene they have that causes this to be yellow is either of the two. Is either it's this and this, or it's this and this. This is the genotype that could cause it to be what? To be yellow. But the genotype that could cause this to be green would only be this gene here, nothing else. So this gene will code for it to be green, but these two genes can code for it to be yellow. These two genes is a genotype. This one is a genotype, but the phenotype is a yellow color and the green color. Is that clear? So the genotype of yellow seed plant could be this or this. But the genotype for the green seed would always be this because it is a recessive allele, not a dominant allele. So let's take a break here. See you later.